So we'll formally transition to the panel. So we're now into our last hour here, where all three speakers will come on for questions. And I've collected a few questions that I see cropping up that I think would be good discussion ones here. Karthik, Mike, and Simon, you'll be live for these, and you can fight for who gets to answer first if you want. The first question is, and it was upvoted quite a bit. So there are differences across the tools. What would you recommend pragmatically for people using different software and deem a sequence viral if it's identified by one or multiple pieces of software? So if you three want to comment on that. We'll start with Karthik, if you want, since you're first.
Sure. I think, to be honest, there's a good amount of benchmarking, and every tool is really different, right? So every tool comes with its own sets of biases that are coming from the references used, the databases used, the training algorithms used, and so on. What I will say is there's enough, benchmarking and comparisons made today. So if you want, if we, at the end of the day, if you like a particular software, just go with it. Just go with your gut. If you have the kinds of questions that you can ask with Viromics data, I don't think you're going to go wrong with. At least if you use a few sets of tools, you're not going to go wrong, whichever tool you choose. If you do want to use more tools, great. If you have the time and the resources to do it, you can use all of the tools. The data is going to increase, but the amount of time and resources you put in, the extra data you're going to get or information you're going to get is just incremental. So that's just my 2 cents, really.
Yeah, I think, I agree with a lot of that. And some of it, I think, depends on how big is your data set, because if you've got, you could do a really great study with, 10, 20 metagenomic samples, and then using a variety of tools on all of those data sets doesn't seem unreasonable. Probably have, virus predictions in the dozens or the hundreds, and you can kind of look at all of those individually. I think I would actually,
I'll just jump in. Like you're making all the PhD students, you're doubling their PhD length by saying go ahead and use all of them.
Oh.
And look at them all. So
don't complain.
One strategy, right? I mean, there is an aggregator tool out there, at least in preprint, the phage. And so I think we should just go ahead and call that out. You could use a tool which aggregates every single one and just adds them up. But I think hopefully an important message you got from all three of these talks today is predicting and putting a confidence on the prediction are two different things. And the tools that we've highlighted here, we've really tried to bring in that idea of confidence, and that not all predictions are equal. And so, these tools really have put a lot of effort into confidence, and that's important, because I wouldn't trust adding up, 12 different tools worth of stuff as right, because there could be low confidence predictions made by a tool with different sets of assumptions that are wrong. Yeah. And so unless you're planning to manually go through them all,
which you should maybe.
So because you suggested they should use them all, I felt I had to jump in.
But I would also counsel in general against using only the union of all the tools. So if you use 4 tools and you only use the contigs that are predicted by all 4 tools, you're actually probably leaving a lot on the table as well. So that's, gets confidence, and maybe Karthik in the end is right, where you just gotta choose 1 or 2 and go with it. But yeah, well, here's something I might think is it might be more okay to use all the outputs from multiple tools if you're using the settings on those tools that are more stringent and doing a better job to remove the low confidence predictions. Like I think I was looking at the VirSorter 2 documentation, and there's like requiring hallmark genes, for example. And with Vibrant, you could use Virome or not Virome, and my tool has some options as well. And so maybe that's part of it. I don't know what you guys think.
I think you bring about a great point. It's important to understand how the tools function. So if you are I think it's important to think of yourself as a casual user or a more dedicated or expert kind of user. So if you think you're going to use this, for one study and not use this again in 2 or 3 years, do you have the time or resources to go in and look at 5 or 6 different software, try and understand how they actually function, what their biases are, and then make an educated decision? Or do you just go off of, other manuscripts that have used one particular software and have, and there are some implicit assumptions that, they've figured it out. So it really depends. I think, my advice generally to like casual users or students, who are not gonna say be able to compare, a lot of different tools, is just go through 1 or 2 papers, and if you think this software is performing well, and of course you can go off of other people's experiences too, then you should just go with it. I want to agree with Matt. It's just that I want to make sure my students are graduating on time, not prolong their PhDs.
Simon, did you want to comment on this first question?
Okay. I would say it doesn't matter, and then I will explain. But the bottom line is it doesn't matter that much per se. You should pick out any tool or combination of tools based on what questions you want to ask, and then most importantly, your interpretation of the results should be consistent with the strength and limitations and caveats of the tools you used. Like Matt was just saying, let's say you use 20 different tools and you took the union of everything, meaning if a sequence is predicted by one tool, you will count it as viruses. Why not? There is some justification for doing this, but when you interpret the results, you have to be aware and be upfront that this is a very permissive way of predicting viruses, and most likely than not, you are looking at a dataset that is enriched for viruses, but contains a number of non-virus sequences. At the opposite end of the spectrum, you could go and do the other side, like let's run 20 tools, and I'm only looking at the sequences that are predicted as viruses by every single one of these tools. So really just the intersections of every prediction tool. That's a much more conservative approach. Again, some great reason to do this, but in your interpretation, you have to be aware and be upfront that this is a pretty conservative way of doing this. So you may be missing viruses. That's all there is. I don't think there is a way to tell, no, you should do this or that. It really depends on what you want as a question. You want every single virus sequence, including some extra, because that's what your analysis requires, or do you want to look for, something like auxiliary metabolic genes, meaning host genes in virus genomes, in which case you must be much more conservative with what you call a virus if you don't want to spend the next 2 years of your life sifting through false positive and be disappointed, one gene after the other, saying, oh, cool, it's in a virus. Oh, no, it's not actually a virus. Oh, this one is, yeah, I've gone through this. So that's speaking from experience. But basically, that's really what there is to it. No combination or no combination, like Karthik said, it's a I think combination has its strength. The caveat is just the amount of time you can sink into it. So it's a balance between how perfect you want your data set to be, how comprehensive you want it to be, versus how much you want the science out of it. And I've never looked at a study so far and thought, well, yeah, they didn't use like the tool I like. So no, we all know the different tools have different strengths and weaknesses. We designed our tools for a reason. It doesn't mean that we just dismiss completely like the other tools that came out, especially if these tools have been published and benchmarked properly.
I wonder, so Simon, to bring that around a little bit as well to kind of what you were saying earlier about very large virus genomes, and I feel like this is a little bit related, is, as I become, I don't know, maybe more curmudgeon-y or something, like I kind of really like a quality check using like CheckV that estimates completeness, because, and obviously no one presented on that today, but you guys should check out CheckV if you haven't already. It's a really nice tool. And I think like I almost don't care if you have a sequence that's not at least like 20-30% complete, because you really can fall. That I think these low completeness also correlates with like low confidence, and I think that can really get you into trouble, especially when you're trying to make a claim about like a really large phage, or it's just like a string of hypothetical proteins as you make them fall. And so I think maybe that is another consideration that comes to mind.
Definitely. I mean, again, like when Stephen came up with CheckV and he started to develop this, I think this is one piece that we are all missing. We were all developing kind of new detection tools. And Karthik had already done this a little bit by kind of fitting into Vibrant, but I like also the fact that it's like a completely, separate tool that you can add to any kind of virus contig collection. One caveat I will say about CheckV, it's reference-based to an extent. It also uses like circular contigs, kind of carefully curated as like representative of complete genomes, which works fairly well, but it will work very well in quote unquote known environments. Like CheckV works wonders in human gut phages, basically. It doesn't work as well. Like you will have a lot of like this NA, like I can't tell you the completeness for like soil or even some aquatic viruses. And so it will get better with time. But for now, I would not say like I'm confidently doing like a completeness threshold and just dismissing anything that is like non-predicted as completeness, because a lot of these are really good sequences. I'm at this point, I know personally, I'm still doing a bit of a hybrid of like length cutoff and completeness cutoff both. But I fully agree that conceptually, Mike is right. Eventually, we would like to go ahead and just like, same way that microbial folks do, just dismiss this low quality, low completeness, low confidence sequences and just focus on the great like near complete genome that we have, and we start to have more and more.
If I could follow up and ask you each to be honest about your tool, which viruses do you think you're working the best with, and which ones do you feel like, gosh, that's a real Achilles heel at the moment? And you're welcome to comment on, oh, but I'm working on that or something too, just to give this students and postdocs some sort of guideline that today they want to run their new virome or metagenomes through to predict viruses. Which ones will they want to go after? Any of you can start.
Well, I can go first, and I'll mention some of the caveats. With Vibrant, I think it performs best on double-stranded DNA viruses. Vibrant does not do very well on viruses that infect eukaryotes. Okay, so I'll be upfront and say identification, and also be upfront with say genome quality. So with regard to Vibrant, when we developed it, we had these ideas of what we wanted to do. So that's why we had genome quality estimates. But we will be upfront in saying that CheckV does a much better job than a Vibrant. With Vibrant, it was designed to just go along with your genome identification and just give you a sense for what you're actually seeing. It wasn't meant to go in the depth that say CheckV did as a standalone tool. So we ourselves use CheckV for those sort of estimations. But as Simon said, we also do have a sense of what we're looking at when we manually look at something. So we sort of mix and match our analyses on that front. So yeah, as far as performance goes, double-stranded DNA viruses, Vibrant does great because our training data set is biased towards double-stranded DNA viruses. As far as eukaryotic viruses goes, microviridae RNA viruses, it doesn't perform as well. For example, with microviridae, we'll always end up calling them as low quality viruses. So some of these biases, like over time, we will improve upon it as we have future iterations of Vibrant, it'll get better. We still are dealing with some plasmids, showing up as viruses. But over time, as future iterations come by, I think, we'll try and iron out more and more of these issues.
Yeah, so I would say Cenote Taker 2, on the other hand, I think probably performs best with single-stranded DNA viruses and RNA viruses and small double-stranded DNA viruses, because what Simon was saying a little before, that actually the density of information within those genomes is very high. The microvirus capsid takes up like 1/3 of the genome or something like that, or 1/4 of the genome. And it's only that capsid is not found anywhere else other than in microvirus genomes. And so that's one of the strengths of the hallmark model, as opposed to something else, I think, is that, when even if you just have a fragment of the genome in an information dense small genome, it's going to perform very well and you're going to miss very few fragments. I think I wouldn't use Cenote Taker 2 on archaeal viruses in general. I would just, they're just so weird. And I think half the time I can't even figure out what the capsid is on those things. And so I wouldn't use it if you had an archaeal virome project, probably. And also it does not work for retroviruses. I should have maybe said that earlier. It doesn't work for retroviruses. I find them too difficult because there's too many endogenous retrovirus elements everywhere, and I just gave up. And I'm not working on that. So that's my answer. But I actually think it does work very well on double-stranded DNA viruses as well. But it's not going to find these little sandbox regions that some other tools might do a better job on.
Okay, for VirSorter 2, same thing as VirSorter and even Profinder behind or before. It's really good at novel phages and actually novel viruses in general, as long as it finds some anchor in the hallmark genes, and then it's really good at looking around and making sure that the genome context around these hallmark genes looks like a virus. So that includes like pretty big like phages. Usually VirSorter 2 is actually not too bad with archaeal viruses, even kind of the exotic ones we have tried to capture them. That's kind of the strength is here. Big limitations, we have the same as Mike, meaning we went into eukaryotic viruses, but we didn't went as far as like retroviruses and stuff like this. This is just like absolutely messy if you start to try to process a eukaryotic genome with the idea of finding all retroviruses. You have so many retro elements and non-viral mobile genetic elements that it's, yeah, we didn't go there either. And then the other part, which Mike also already illustrated, is prophage boundaries. It's not something we have pushed very hard on VirSorter 2, parts because of a lack of time, and parts because frankly, I'm still struggling to understand how we can better define these boundaries. We have collectively made incremental, I feel like, advances in this realm, but I'm missing kind of the next step where we'll finally get from like, it's marginally better, but it's still not great, to like, oh, okay, yeah, we can finally define this. Maybe we never will, I don't know. But like prophage and provirus is definitely something, and specifically the exact boundaries of this prophage prediction should not be blindly trusted coming from VirSorter 2 for sure.
Yeah, to piggyback on that, I don't know if we're ever going to figure it out per se, because based on sequence information alone, because oftentimes there's an integrase at one end of the prophage genome, but actually a lot of times there's one or two genes even more terminal than that. And on the other side, and then there's also oftentimes integrases within the body of the phage genome. And then, oftentimes the other end is like this part of these sandbox regions in which things are coming in and out all of the time. And there's even other mobile genetic elements integrating in there. And that's just a whole nother level of confusion. And often, some prophages are like these grounded prophages where they're actually not able to excise and make particles anymore. And those are kind of hot spots for all kinds of things coming in and out. And then that even confuses it further. So it is worth knowing about the challenges of those.
And I would add one more super quick, Matt, before, but it's really quick. It's prophages, a lot of the time, or at least a number of times, prophages will integrate into regions that are themselves kind of hotspots for different mobile genetic elements. So it's not like you have a very clean way of like a prophage integrating into a middle of like a core genome, and that's great. But then you have like a prophage integrated next to a prophage, integrated next to a transposon, and then you have like 4 back-to-back transposases, and like where does it start, where does it end, what is the unit of the prophage, who knows?
Well said. Yeah.
Great. So related to prophagens, and then I want to come back to the first question again, just to make some comments. Related to prophagens, I just want to flag for all of us as a field, this is the huge risk with AMG studies. Those boundaries, as soon as you have an interesting metabolic gene, it's not interesting if it's not in a virus, right? So Karthik's got a set of rules. I put some sort of user beware links to some of Simon's past work, and Ajit Pratama's put out some standard operating procedure rules. So those links are in the chat. But I want to make sure people realize that, just because VirSorter 2 or Vibrant or Cenote Taker calls it as a virus, you still want to check the proximity around it, because it could be in a prophage region where the end is not called well in particular. And then back to the first question, we're at a stage, this is a nascent field. A lot of the tools are being developed sort of in real time. In fact, a number of these tools came out within a couple of months each other. It was great to see all the activity. So they were really, truly independently developed. And so So a next step as a field could actually be, I see that each of these tools here and a number have a curated database of, virus-like proteins scored in different ways. And so an opportunity might be for us to come together and put in for some funding for a workshop to think together about creating a centralized database and even maybe and what chassis we want to plug into, or maybe we can put together a couple of chassis if we feel that there are strengths to be realized on both sides. So I just plant the seed. I don't know, you're all welcome to comment on it, but it reminds me of a number of years ago, the first grant that I got was with Maya Breitbart and Jeff Elhai and Rob Edwards. And the crazy task we were trying to do is work with a bunch of phage biologists to just annotate better on RAST, the Rapid Annotation Subsystem server. So it's, we're at that point where we could write a kind of community effort and create the shared resource that we'll all benefit from immensely. Maybe they'll be part of the database discussion on Friday.
Well, then what will we argue about?
Oh, I've got a long list.
Okay.
Do too. I'm mostly thinking about students, and the first step in any of this work is virus identification. So what I don't want to see is a student losing 9 months troubleshooting every tool under the sun because the benchmarking isn't systematic, or some tools don't have this. I like the comments here that, hey, just use a tool that's well benchmarked and move on, or use a couple of tools that are well benchmarked and move on, right? You can focus your energies in other places. That's the message we want to put out there.
I might even come out as anti-benchmarking to some extent, though, because any benchmarking that you do is going to be kind of filled with the biases of the field or of yourself about what you use for a training set, what you use for a test set. It's actually, I think, extremely not straightforward. And if you can, not that I'm excusing anyone doing this, but you could have the best prophage prediction tool that predicts all the E. coli phages like no tomorrow, but then it might totally fall apart once you get to some other, taxa or something like that. So it's, I don't know. I mean, I think probably people are smarter than me to figure it out, but I find it very dizzying.
And I think part of that discussion, so if we were to develop a curated set of underlying viral-like genes, part of that discussion is also in that space too. How are we going to benchmark? What do we all think are the pros and cons of this way, that way, and the other way? I think it'd be great to work together. If it's okay, I was going to shift to a slightly different topic, unless you all wanted a parting comment on that first round. Okay, great. So the second topic is, we sort of touched a little bit on the kinds of viruses that your tool may work better or worse for. I wondered if you could comment about the kinds of ecosystems. There are questions about, can I apply this ocean benchmarked system on human gut or lakes or soils? Maybe you could each go around and just comment on what you think about the different.
So I guess I can start. When we designed Vibrant, I think we wanted to ensure that it's working well in all environments. But that's say how we had the vision in our head. In our mind, we then tested it on other data sets and saw that it was performing well across different environments. But I think we are just in our we are finding bubbles inside of our bubble somewhere over there, because I think it's just biases that are coming into the play. I think depending on the data set, or say the references you use, so to train your algorithm, or say how you would find viruses, I can imagine that some virus finding software work better in certain environments. I mean, it has to be like that. But in my mind, I think as far as Vibrant goes, if you understand the biases of Vibrant, if you're comfortable with it, then you're going to find similar results and you'll be happy with those results across different environments. But I'm sure, we are not doing as well, or we are working against some kind of viruses in certain environments. So it's a very ambiguous answer, but that's honestly the best I can do, really.
Mike or Simon, do you want to jump in with a comment?
I don't know that I have much to add there. I mean, yeah, I think different ecosystems are going to have different representations of taxa, and so therefore different tools will perform differently. But I don't know, man. That's all I have to say.
Yeah, it's pretty much the same. Adding to ecosystem, like you actually did, Matt, in the early intro. The way you process your sample will heavily bias whatever viruses you will see in your dataset, and that should inform your choice of tool and interpretation eventually. Said otherwise, if you start from a very low input sample and you are interested in single-stranded DNA viruses, go ahead and run some MDA. You will get plenty of single-stranded DNA virus genomes, and that's great. You can't really claim anything on double-stranded DNA or other viruses because you would have skewed your data sets towards single-stranded DNA viruses, for instance, which is not bad per se, but it's, again, something to always be mindful of and upfront with when we present this kind of results.
I think one easy way I can sort of, at least in my head, I can think of say introducing bias into your software or say your results is using, let's say, a very limited data set. So if you use, let's say, an NCBI reference data set, then it's heavily skewed, let's say, towards cultivated viruses or viruses of pathogens are coming from humans, then you're definitely going to skew all of your outputs. You're going to skew your algorithm. So if you determine that the algorithm has used a very representative data set and that's sort of what you're looking at, then I think you would be happy with what you find. So.
And a little bit related to that, someone was asking about whether sequencing technology might matter, and does one tool or the other perform particularly well on short read versus long read data, Nanopore, PacBio.
Here's what I could say about that is like PacBio and Nanopore to some extent, but more so Nanopore, they do have these indel issues in the assemblies, and which means it's more likely that a hallmark gene is going to be artificially, broken in half or something like that. And so instead of finding one hallmark gene, it might find two separate hallmark genes for the same major capsid protein, for example, right? And so you might be overconfident in the call if you're just looking at the number of hallmark genes that are called. On the other hand, if the ORF is broken in a particular location, that might prevent the actual detection of the gene family hallmark in that ORF. Another thing is looking at circularity, the way that Cenote Taker is currently set up. Unless there's probably more assemblers I don't know about, but the long read assemblers that I know about won't usually leave overhangs that you can align back to each other. So circularity probably won't be detected. Though of course you could look at the assembly metadata to figure that out yourself, I suppose. And then of course you could have also, on the other hand, you might have very long direct terminal repeats that are present on the end of a virus genome that Cenote Taker might actually think is the end of a circle, and so we'll circularize those instead of leaving them as actual direct terminal repeats, which they might be.
Yeah, I'll agree and second what Mike said. In my mind, we didn't design Vibrant to depend on a particular sequencing method. It's supposed to be sequencing method agnostic, but what comes out really depends on the depth of sequencing, quality, so many different parameters. So if you use different sequencing methods, I can imagine there are differences, but we never checked for this. We just assume that you are presenting the highest quality assembly possible, and then we can tell you something on the basis of that.
Yeah, if you work from assemblies, it doesn't really matter what your underlying reads were. And for all of the tools we talk about today, and for the vast majority of the tools that are out there, you should work from assemblies. I don't know of a tool that reliably works for reads outside of the tools developed to find known viruses, like more clinical applications, for instance. To find novel viruses from reads is something that is very, very tough. I've not tested it extensively. anecdotally the same way as Karthik, I think, was just hinting at. We have seen this issue with some assembly from long reads having homopolymers and indel issues, which means your gene prediction is all kind of wrong. And that means that in this case, Camerabased tools may be better suited to kind of deal with that, although I have not, again, tested it very well. And I would say it doesn't matter, because even if you can detect your virus sequence, if you have a quote unquote crappy assembly, you can't do much with it. So I would say clean up your assembly before actually trying to find virus sequence, in there. But otherwise, yeah, we have used Viromics on a number of kind of recent PacBio metagenomes, for instance, and it works very well. Again, because we have this recent PacBio where the base quality now is on par with Illumina, and so you get very good assemblies. And at this point, it's the same as any other assembly data. And I think Ben, which is one of our experts on long reads.
I see one of the panelists. Yeah, go ahead, Ben.
Yeah, hi. So I was just going to say, we initially tested Camerabased approaches on the long read data when it first came out, and we took an E. coli genome that had been sequenced in long reads, and it has had basically a 97% false positive rate for viruses in there. It was just almost every read it assumed was viral, because the Camers looked so weird because of the kind of the homopolymer issues and everything else. So
amazing.
I would definitely not recommend Camer-based approaches. For
thank you for correcting me, Ben. I'm glad I say I didn't test it, because yeah, apparently if you test it, yeah. Perfect. So yeah, if you have like this kind of issues with your reads and your countings, I guess there is no tool that can bypass this. You have to clean up your assembly before doing anything.
Yeah.
And so, Ben, can I follow up? Is that just on the reads, or after you did assembly, did it still have that same issue that you're aware of?
This was purely on the reads.
Okay.
So yeah, I mean, these days with the higher quality of the reads that are coming out and the better assembly tools for, tricycle and unicycle and these kind of tools, but they're getting better at polishing. And so actually after the polishing, even without kind of short read polishing, the difference in the lengths of your predicted coding regions is actually, it used to be like your long reads had really, really short coding regions, and your short reads didn't. And actually the difference between those two is now smaller than it used to be. I'm still finding that the long read only assembled stuff still tends to have slightly shorter predicted coding regions because of those indel issues, but it's improving in the right direction on that front.
I cannot believe I'm going to say this, but this conversation is really reminding me of 454 days when predictions were really difficult.
You just dated yourself, Karthik. Now everybody's Googling what 454 is if they're newer to the field. Great. I think there's two other venues or two other sort of discussion areas I'd like to kind of get at, reading between the lines in the question. So the next one would be coming back to eukaryotic viruses. And the comment is, it seems like eukaryotic are an afterthought with many of these tools. First they said, thank you for all your amazing talks. And then they said it's an afterthought. What do you think the future of eukaryotic virus discovery is?
I would say eukaryotic viruses are not an afterthought in Cenote Taker 2 at least, but they're also like, maybe I'm going to embarrass myself by saying something wildly inaccurate, but I feel like the diversity of eukaryotic viruses is approximately like 1 or 2% of compared to phage diversity. And so it's impossible not to spend the vast majority of your time and energy thinking about the phage sequence space when you're thinking about virus detection in general. So I'm sorry in advance. But I do think that, even so, I'll say, like, there was a cool paper that came out like a year or so ago in PNAS about this really, really weird amoeba virus that was called, what was it called? It was called, I have it somewhere, Yara virus. It said like, oh, all of these open reading frames on there are like totally undetectable. And Cenote Taker 2 was able to find it just fine. Probably some of the other tools as well. And so I hope that for the most part, it does pretty well. I mean, it's not going to, but again, my tool will not find little short fragments of things that are not virion hallmark genes or something like that. So where's the future going? I don't know how much of a future there is, I guess. I feel like it is just, I mean, I could be totally surprised, but I feel like there's not going to be a tremendous amount of diversity that's yet to be explored.
I guess to add to this, there are a few reasons in my mind why you feel like there are new tools all the time that comes up to find phages and everything, and less for eukaryotic viruses. And I think it's a weird thing with eukaryotic viruses where it's either quote unquote a solved problem or it's impossible. Let me get into this very quickly. Phages have a super broad diversity and everything. They have plenty of novel phages out there. And detecting them, these new phages are usually somewhat very decently related to your references, enough to be a challenge, not too much to be completely impossible. And so that's why people like to explore phage studies. In eukaryotic viruses space, you can go from very short, small viruses and small genomes. These ones, basically right now, like Mike just presented Cenote Taker that is especially well geared towards these small viruses. They have well-defined hallmark genes. Again, they will come out as almost systematically complete assembly from your metagenome. So from a methods point of view, we don't have a lot of tools that are very well done to kind of wrap up everything around, again, before we had Cenote Taker, but we can find them relatively easily. A lot of eukaryotic viruses are also RNA viruses, and RNA viruses have this wonderful feature of having this, by design, or not by design, by default, they have to have an RDRP. And that's, compared to all other viruses, you have this unique chance of having this hallmark gene that you can recognize by using what is now a pretty expanded collection of HMM profiles to find this functional domain. And that kind of sign in your metatranscriptome, what is an RNA virus and what is not. And then we can talk about segmented virus for RNA viruses. And that's where you go into what I call the impossible. Like finding the RDRP containing segment is quote unquote solved. We can improve on this, but it's not something extremely new to do. Finding the part of the genome, if it's a segmented genome, meaning it's in different templates and different independent segments, finding the segment that doesn't have the RDRP right now, as far as I've seen, is nearly impossible for new viruses because these segments are short. They usually encode like new proteins, and you will have tons of like short contigs with uncharacterized genes in a meta T, and you don't have anything to go from the sequence itself. And so that's what I meant when I said like it's going from like quote unquote solved by using marker genes, some like things like this, to nearly impossible. And then we can go all the way to giant viruses, and you have to use like specific, like I was saying, you are using kind of a hybrid with like genome binning and stuff like this. We don't have a good tool to automate this yet. I think that's maybe where there is some work that can be done that can, eventually lead to a much more robust and systematic way of getting this kind of, NCLDV from metagenomes. But for the shorter genomes, yeah, I'm like, we'll have better HMMs, so we'll have better way of finding these hallmark genes. And then for the sequences that are nearly impossible to distinguish either from, completely new stuff or like the retroviruses, like we mentioned earlier with Mike, we'll need a big advance to be able to tackle this.
Now, the only thing I would add to what Simon said is that, to be completely honest, eukaryotes right now are an afterthought in most environmental and human microbiome studies. So if eukaryotes are an afterthought, then eukaryotic viruses, you can imagine, are also an afterthought. I'm not saying it's the right thing. It's just how it is. I think more and more people are studying protists and microbial eukaryotes now. So this will only get better, but I mean, that's just a general, just the general status of the field, to be honest.
Mike, do you want to reply to that? I'm looking for anything controversial here.
Very good points. I mean, that makes me think also of like fungi, which are pretty prevalent in metagenomes or environmental samples and stuff like that. But then their viruses are like, what are their viruses? They have like very few somehow, and they're like half of them are like metaviruses, which are they really even viruses? And then I guess there's some like crest viruses that they have.
A little bit of that, a bunch of double-stranded RNA, which again are manageable with RDRPs, but once you get out of these, metaviruses are also a pain to find. Yeah, it's really kind of challenging.
Yeah, I think if someone was motivated, they could develop probably a tool to find new retroviruses that would fill a big niche. I kind of volunteer to not do it. So if anyone wants to do it.
Great. Okay, so another question is going to get us into the viral matter space. So what if we identify a phage sequence based on only 1 to 2 hallmark genes, but all other genes have unknown functions? Is this really a phage? Is it even interesting to look at? We still know very little about it. I wondered if any of you wanted to comment on those borderline dark matter cases.
I was actually just writing a written answer to this, so I'm so glad you chose this one. You can count me in the camp of still confused about the definition of viral dark matter. I'm straight in there. I used viral dark matter in the past. I'm guilty of this. I'm trying to stay away from it because when you think about this more deeply, the phrasing doesn't make any sense with what physics dark matter is. So I'm trying to get away from it. More generally, yes, finding a virus or being able to say, oh, here I have a new phage genomes with 3 gene characterized and 20 uncharacterized, by itself cannot be the end. It's only the beginning. There are plenty of things you can do downstream, but it's really your new genome is basically the start of many downstream studies. And then what it lets you do, it lets you focus, because you have so many different phages, so many different genes that are uncharacterized. How do you go after the ones that you think are the most important? And having these genomes lets you do this, because it lets you do protein clusters and understand, okay, what are the genes that are shared? What are the genes that are conserved? What are the ones that are found in all of these environments? And then it lets you do read mapping to find, which virus is abundant where, which also lets you tell, okay, maybe this gene is important there. Also, it lets you do gene neighborhood things like, oh, here is a gene that is involved in a pathway that I really like, and it's surrounded by 3 unknown genes. I guess, maybe one of these 3 may be important there. So again, it's only a starting point, and it's here to help you decide where you go next with your more targeted assay and experiments, but definitely not the end all be all, even though a lot of studies, stop there for a lot of different reasons we probably don't want to go into today.
Yeah, I mean, to some extent, all these hypothetical proteins are just they're labeled hypothetical proteins, so you have to put them into AlphaFold, right? That's why they're labeled like that.
And then you get a pretty structure, and you have no way to know if this is a correct structure or if it's doing anything. So,
but it's a structure.
But at least you're in structure.
And then it's solved, right?
It's pretty, yeah, that's a good point. But it's a bit of the same issue, right? It's like you get a genome, cool. Where do you go from there? Yeah, we have a lot of work to do from there.
Yeah, but to expand on that a little bit, and I don't know if this gets to the heart of the question, but I do think, and there's been a couple or a few papers that I've looked at kind of viral pan genomes, like the variation in gene content within a viral species. And I think of some more information you can get about all those hypothetical genes is how constant are they in the species, or are they these genes that are getting moved in and out? And that's not function, but that is as a clue about something at least. And I mean, was part of the question, how do you know if it's really a virus? I think we kind of maybe, I don't know, have we belabored the point about how do you trust the outputs, things like that. So maybe I don't need to go into that again, but yeah, I mean, and also, if you're the one who's going to figure out all of the functions for all of the hypothetical proteins, that's like, everyone's going to love you. So you could say it's a good thing that we don't know, because that's, something else to do. That's very exciting.
I'll also just add a little bit of a philosophical touch to that. I mean, if it's a virus, great. If it's not a virus, still great. I mean, it's full of hypothetical proteins. It's full of unknown proteins. Unknown today does not mean unknown tomorrow. Someone will solve the actual structure for that protein, or someone will find an actual function for that protein. And then you will finally have context for your specific virus or whatever that sequence was. So,
hang tight and stick with it, right? If you like it. Yeah, great. Thank you all for that. There's another one here about I'm going to call it the interpretation side. It's a little related to this. So one person's noticed a huge difference between FASTER and Cenote Taker's results. What should I keep as a prophage when only one sequence overlaps out of 15 predicted? Do you want to give sort of the sort of logical rules that you use to curate?
Well, I mean, part of it is going to depend on which settings used in Cenote Taker 2. So obviously either FASTER is very relaxed or use something strict on Cenote Taker 2. So if you're doing like a bacterial genome, I think you can just use the Virion database, and you can do 1 or 2 hallmark genes depending. I think 1 is probably fine to require it. But I mean, I don't actually know a lot about FASTER. I mean, my guess, to preserve my own ego, is that FASTER is giving you 14 false positives. I mean, I don't really know. I don't actually know about what FASTER actually does, but or is it that Cenote Taker 2 is giving you a bunch? I mean, I guess without more, I mean, I struggle to answer that without kind of more info, but if you want to send me the sequences or send me an email or a GitHub issue, and the problem is Cenote Taker 2, I'll definitely would love to be aware of it. I don't feel like I see a lot of false positives using the correct settings with Cenote Taker 2. So, I mean, yeah, that's.
Yeah, I think it sort of comes back to one of the original questions. You use 2 different tools and you get different results. Sort of how should a trainee try to figure out what to trust? And so you have some rules that you've built into your tool. And I think a trainee could read the rules and say each of these 3 tools and other tools that they find to be good. Those rules are there for a reason, that a lot of people's eyes have manually curated for years and years and years. And that rule set is the logic we would use to curate. So I would say in this case, these might be regions that FASTER or Cenote Taker handle well in one tool and not the other. So see if you trust the rules that are being used in the 2 tools. And Mike can certainly help you interpret that. A slight variant of earlier, I asked you to sort of put your vulnerability out there with respect to different viruses you might not be tracking well. A slight variant is someone asked about structural features. Are there particular classes of gene sets you don't think we're handling well yet as a field or in your tool in particular? Do any of you want to comment on that one?
Hypothetical proteins?
That and short proteins. Short proteins are a pain. HMM profiles are, I was going to say godsend. It's maybe a bit strong, but they are amazing at taking multiple alignments and condensing the information and really like getting the meat of it, meaning getting the conserved residues and letting you kind of find related proteins to kind of this protein family. And you can do them at a number of different granularity levels. You can use pretty sensitive tools like MM62 and stuff like this, kind of HH Blitz, and really go far in terms of protein space. But you do need sequence information for this, meaning you do need a proper functional domains and a relatively large sequence. So it's hard to do for hypothetical proteins because you are all basing yourself from the sequence. And so you're like, okay, yeah, this residue and this residue are conserved, but you don't have any way of knowing that these are actually functional and important. It may be just artifactual. And then it's really hard to do for short sequences because, and short proteins, I should clarify what I mean, roughly 30 amino acids and below, or maybe 50 amino acids and below, this range basically. We know they exist. We know there are some genuine CDS and proteins are in this length range. They are extremely difficult to predict in silico because if you predict protein in this range size, you will have a lot of false positive because you will have a lot of ORFs that exist in the genome that are not, actual CDS. And then building HMMs from them, just because your alignment is so short, it's really complicated. So I would say, yeah, hypothetical proteins and short proteins are probably the blind spots here.
So I agree with Simon, and I'm just going to add like a little bit of a vulnerability, as Matt mentioned. I think one thing that we've struggled with is when the way I think most software predictions for viruses go is you, at least if it's gene content tools, you're using a standard gene caller that is the same for viruses and microbes, and you're basing some sort of predictions off of that. Now, if you've identified a virus for sure, you can go in and refine the gene calling after that. But you can't go in thinking, you have a very specific, very virus-specific gene caller. And I think that is something we've struggled with. I don't have a good answer. It's just a question as to, what can we do about this? This is a question that constantly bothers us as to whether when we call something an off, how confident are we because of just how weird and different viruses are, and even viruses from their different families are.
Yeah, so there's, Karthik, have you seen this tool called Phanotate?
Yep.
Yeah, and so, I don't know that I'm sure it's not perfect, but it does, I think it does seem to do a better job at finding the smaller ORFs within viral genomes.
Correct. So, but my question is, so Phanotate will work on viral genome. So for example, if you've identified a sequence as viral, then you can go in and annotate it using Phanotate, or say another phage or virus specific off finder. But if you have a mixed metagenome that contains virus sequences and microbial sequences, and you're doing off prediction on all of them using some sort of a baseline, function, how good is that? We think it's fine, at least for calling phages. That's why we think all of these softwares even work, right? But it's something that internally we are continuously debating.
Yeah, I tried I guess a halfway method with Cenote Taker too, where after I've identified a sequence as a putative virus and then trimmed away the non-prophage regions if necessary, then I use a step to just like a blast X quickly to try to sketch out if I think it's a phage or not, and then I'll use Phanotape just on that region of that individual config if it's a phage. But if it's I think it's a eukaryotic virus, then it just gets Protaville meta. So that's maybe slightly better, but it is tough because you can have different genetic codes in your metagenome and all kinds of things.
The Phanotate doesn't scale for metagenomes, so I know they're working on that, but it's not currently an option to use in all contigs. But it is.
Yeah, I run the contigs individually through it, which is slow,
right?
Probably, but
that PhD just lengthened again. No, but it is going to be great for the specific questions. So, but I know they're working on a scalable version too, so that's good. I want to come back to another question. I realize it's a little bit different. We only have a couple minutes left, but someone asked about MAGs or metagenome assembled genomes. Would anybody want to comment on sort of how should those be specially handled? Any concerns there? Thoughts?
Yes, please. Yeah, I went through this, so I really like this question too. So MAGs meaning collection of metagenome contigs that are supposedly representing one single genome. And usually, as far as we can tell, they are pretty good representation of genomes. You can definitely use any of the virus detection tools we mentioned to find virus region in there or virus contigs, and they will work in the sense that you will find virus regions and virus contigs. Now, the next question is, does finding a virus contig in a MAG tells you anything about this virus infecting this host? There are two answers to this. One is yes, if, and that's a very big if this virus region is on a contig that's also includes a pretty big host region. Pretty big meaning several KB, 10 KB and more. If you have 10 KB or more of host and then some virus region and then whatever, then you can trust the binning, or in our benchmarks, you can use this binning information as some sort of host information. Not perfect, but it's there. What you shouldn't do and what you should be very careful with is contigs that are completely viral binned into a microbial bin. This is absolutely not an indication of host. Completely viral bin contigs will be binned completely randomly with anything. And that's because the GC content and the coverage will not match between the virus and its host, basically. So if you have virus contigs and they are in a genome bin, don't take this as a host prediction, because this will lead you down the wrong path. And I think Karthik went through this as well. I'm sure you have seen this.
Yeah, I agree 100%. There is not, I can't, I mean, you've put it together very well, so I don't think I need to add anything on top of it.
And one thing related, Simon, I'll put one of our vulnerabilities out there. We tried some genome binning from GOV1, like global virome 1 data set. And then when we got more sequencing data, we actually saw that a lot of the binning on the virus contigs wasn't good. And so if somebody's asked in here about binning,
I was going to say that's a slightly different question, but that's probably good to address as well. What about viral genome binning? My experience, and again, your mileage may vary, and that may be just my bias, but I don't use viral genome binning except for very big genome, like when you start to go in this megabase range. Below this, at this point, by the time I feel I will have a proper genome binning tool and everything working very well, I feel like we will have assemblies of like complete genomes coming straight out of the data. So yeah, I'm not spending too much time thinking even about this, which may be a mistake, but yeah, we'll see.
Two minutes left. And I want to stick to time. Thank you all very much. That was really informative. I think that should be really helpful for all the trainees out there who are navigating the virus identification space. For the rest of folks, tune in tomorrow to hear about virus classification tools and Friday virus database tools. Hope you can put your hands together, and thank all three of our speakers today. They did a great job laying the landscape here, setting up the landscape for this first day. Thanks, everyone.